It says live in the corner now.
Okay, and we're live. Welcome everyone to the very first edition of our Limno seminars, Limno seminar series. This was all inspired by our colleagues at micro seminars and philo seminars, and so I encourage you all to check out their website. They've been a fantastic help to help us launch this seminar series, so a special thanks to Cameron Thrash and Jennifer Biddle to help us navigate the intricacy of just launching this. So thanks a lot. My name is Alexandre Poulain. I'm a professor, associate professor at the University of Ottawa. We hope we can make this a monthly series dedicated to freshwater and I mean I should say limnology, so study of inland waters. I would like to first introduce our speaker then. Today we have Matt Bogard, who was the Rob Peters Award from the Society of Canadian Limnologists for this year. Matt is originally from Saskatchewan. He received his BSc from the University of Saskatchewan, where he studied limnology as a summer student with Jeff Hudson. I'm reading my notes a little bit. He did his master's at the University of Regina with Peter Levitt, studying nitrogen biogeochemistry and the eutrophication of prairie lakes. He's currently at the University of Quebec in Montreal working on his PhD with Paul Del Giorgio. His research focus is now on the link between ecosystem metabolism and greenhouse gas dynamics in boreal lakes. Matt is interested in the regulation of CO2 and methane flux in lakes, but will be today presenting exclusively results on methane in this talk. His work is part of the broader lab research interest in carbon biogeochemistry of boreal aquatic ecosystems. And soon, this is very exciting, Matt will be starting a postdoc at the University of Washington with David Buttman exploring carbon cycling in Arctic lakes. So thanks everyone for joining in. Matt, I think you know what to do.
Thanks a lot, Alex. I will share the talk. And let's see here. Okay, how's that? Okay. All right, so thank you very much, everyone, for taking the time to hear this presentation. So what I'll be presenting to you today is a portion of my PhD research, looking exclusively at methane fluxes in lakes, and I'm especially interested in the role that methane production in the oxic pelagic environment might play in supporting methane fluxes from lakes. So until very recently, the role of methane emissions from inland waters was not actually included or considered in many of the global methane budgets. But more recent evidence shows that freshwaters or inland waters, so lakes, rivers, and wetlands, reservoirs, collectively might account for a large fraction of total planetary methane emissions. And this has gone overlooked for a very long period of time, but when we look closer at methane dynamics in lakes, it's not that surprising that they could collectively be an important component of the global methane budget, because as you can see here from a survey of boreal lakes conducted in our lab, methane concentrations are supersaturated relative to atmospheric levels in lakes of all different shapes, sizes, and characteristics. And so collectively, they act as a net source to the atmosphere, emitting methane to the atmosphere. So the methane that is found in lakes and emitted to the atmosphere is understood to be derived from microbial reactions occurring in anoxic environments, so anoxic zones of the lake. And this is well understood because the production of methane is a very energetically unfavorable reaction compared to other processes. and so it's understood to be restricted to zones of the lake that are lacking dissolved oxygen. So the littoral zones and benthic zones, where oxygen can be absent, are generally understood to support methane emissions through diffusion across the air-water interface, as well as bubble-mediated release to the atmosphere directly from the sediments to the lake surface, and this is termed ebullition. So a large fraction of the methane produced in lakes can be oxidized, but still enough escapes oxidation to support methane concentrations in supersaturation relative to atmospheric levels and drive the net outgassing of methane from the lakes to the atmosphere. Now, in lakes that are the vast majority are small environments that are closely connected with anoxic habitats, it's clear that methane produced in these environments can support and predominantly drive emissions. But especially in marine environments and in marine literature, as environments are increasingly larger and larger and larger and disconnected from anoxic habitats, it's been observed that methane concentrations are sustained at levels above atmospheric equilibrium, and these very large open environments disconnected from anoxic habitats still support net emissions of methane to the atmosphere. So studies that have looked more closely at these environments have found that in order to close the budget for open water mass balances of methane fluxes, an internal source of methane is needed to account for the rates of emission observed in these habitats. So the different pathways proposed for internal sources of methane in these oxic water environments include methane production in anoxic guts of microinvertebrates and in fish and other organisms. It's been proposed that methane can be produced on particles and in clumps of material in microanoxic zones. And it's also been proposed that methane can be actively produced in microanoxic zones associated with cell surfaces of plankton, especially phytoplankton. So for lakes, recent evidence is accumulating suggesting that there is a close association between methane dynamics and methane production in pelagic environments and phytoplankton growth and metabolism. So on the left, you can see that in a number of systems, methane concentrations profiled vertically in open water environments sometimes show overlapping peaks that are consistent with peaks of dissolved oxygen and peaks of algal biomass. So at the same time, a survey of German lakes has shown, as you can see on the right, has shown different relationships between methane concentrations on the y-axis and dissolved oxygen concentrations or saturation levels on the x-axis. So for bottom waters, there's a very different relationship between methane and oxygen relative to surface waters. So in these bottom waters are anoxic and hypoxic, and in surface waters there's much higher concentrations of dissolved oxygen, and there's a positive relationship with methane concentrations. And so this is really a counterintuitive relationship based on our conventional understanding of methane dynamics, based on the redox gradient that I showed you earlier. So a closer look at these pelagic habitats has shown that methane is actively produced even when accounting for oxidation. So the net production of methane is positive in these pelagic zones through in vitro incubations. Now, also in these environments, there's a close association and a presence of methanogenic archaea that are closely associated with phytoplankton. And at the genetic level, one study has shown that the production of methane, that these archaea are actively producing methane. So with all of this in mind, we were really interested in what the potential role of this production of methane in oxic waters might mean at the ecosystem level in terms of which are the relevant biochemical pathways and how this contributes to overall fluxes to the atmosphere for the whole lake ecosystem. So to address this question, we used a mesocosm experiment using 1 meter diameter by 6 meter deep mesocosms that were deployed in Lac Cromwell, which is a small boreal lake located at the Biological Research Station of the Université de Montréal, about an hour north of Montreal. So we used this experiment to test the rates of emissions, sources of methane, and the relative importance of methanogenesis in these environments. And so we tracked methane dynamics in 4 different treatments. So 3 mesocosms in each treatment were used, and one treatment included the addition of terrestrial organic material to darken the water column and promote heterotrophic microbial activity. We also had a treatment of nutrient addition, including nitrogen and phosphorus, that was used to stimulate autotrophic growth and algal growth in the pelagic environment. We had a combination of the 2 treatments, and we also had ambient control mesocosms. So these enclosures were filled with oxic pelagic water, and they were given a week to stabilize before the chemical additions were made and the experiment began. So over the course of the 28-day experiment, all of the enclosures, all of the treatments, all of the bags had methane concentrations supersaturated relative to atmospheric equilibrium. And in fact, the actual concentrations of methane increased to varying degrees across the different treatments. It's important to point out here that had there been no internal source of methane production, we would have expected the concentrations to equilibrate with atmospheric levels within roughly a week. So based on the physical conditions in the surface waters of the enclosures, we would have expected a trajectory denoted by the dashed lines for each one of the treatments. But clearly this did not happen. And a source of methane was actually needed to sustain the concentrations that we observed here. So across the different treatments, not only the concentrations, but the actual rate of methane emissions across the air-water interface to the atmosphere were strongly related to net ecosystem production, or NEP, which is denoted on the x-axis. So NEP is the balance of autotrophic growth and heterotrophic respiration. So in the enclosures that were increasingly dominated by algal metabolism and NEP was shifted in the positive direction, methane emissions increased to a certain point. So this was a clear, there was a clear link between algal dynamics and not only methane concentrations, but actual rates of emission that were consistent with the previous studies that I showed in the introduction. So it was clear that methane dynamics changed as a function of algal metabolism and algal growth in the enclosures, but we were also interested in the specific biochemical pathways that were involved in supporting methane concentrations and emissions. So previous work, primarily in marine environments, has suggested that methane production could be associated with the demethylation of organic matter by microbial plankton in the pelagic environment. And at the same time, there's evidence from a freshwater study that suggests that methane production in the pelagic environment could be linked to acetoclastic and hydrogen or trophic methanogenesis. So to get a relative sense of the importance of these individual pathways, we looked at the stable isotope signature of methane and CO2 to get, as a qualitative way to assess the importance of these individual pathways. And so what we found was that as we shifted to increasingly phytoplankton-dominated pelagic environments, the relative contribution of acetoclastic methanogenesis seemed to increase. So this really suggests that, at least during day 7 of our experiment when we conducted this analysis, that the reduction of acetate could be the main source of methane in these pelagic environments, or the main pathway supplying methane. And so this observation was actually consistent with a number of different lines of evidence that we found in the literature. So first, for a long time now, radio-labeled carbon cycling experiments have shown there's a very fast turnover of the pelagic acetate pool in oxic water environments. Recent marine studies have shown that fermentative bacteria that can supply acetate might actually be widespread in pelagic oxic environments. And also we know that the precursors for the formation of acetate are widely available from material derived from different plankton. And lastly, in our experiment, we saw an absence of nitrogen-fixing cyanobacteria. So this really suggested that one of the major sources of hydrogen that could fuel hydrogen-trophic methanogenesis was absent in almost exclusively absent in our experiment. So now, to put this into kind of a whole lake context, we were interested in what the contribution of methane fluxes from the enclosures, from the pelagic oxic environment, might be relative to whole lake fluxes and the surrounding lake environment that was connected to these anoxic habitats. And so what we found by measuring rates of methane flux in the surrounding lake environment and rates of ebullition, or bubble-mediated release, in the littoral zones of the lake, was that averaged over the summertime period, the production and flux of methane from oxic waters could represent roughly 20% of total or average diffusive emissions from the lake, and that the flux of methane derived from these oxic water environments was roughly equal to that of the flux linked to ebullition. So because Lac Cromwell is a relatively small lake with a very close connection with anoxic habitats, we hypothesized that the contribution of oxic water methanogenesis represents really an important baseline source in this habitat and likely other small lake environments, but that this relative contribution will shift and will increase in relative importance in ecosystems as we shift from very small, well-connected, very small environments that are well-connected with anoxic habitats up to very large ecosystems that are disconnected from anoxic habitats, such that in larger ecosystems and in ecosystems that are disconnected from those environments, this flux of methane and the production in oxic waters might be the predominant source. So in line with this hypothesis, the results from our meta-analysis suggested that in these environments that are very large open water habitats, oxic, that are disconnected from the littoral and benthic environments, there's a strong relationship between the concentration of methane in surface waters and algal biomass. So this relationship is absent when we use when we look at whole databases, including very small lakes. And this is exactly what we would expect. We wouldn't expect a relationship between these variables in environments where methane dynamics are primarily regulated by what's happening in anoxic environments. But Here in these very large ecosystems, there's this clear relationship between the amount of methane in the water column and the amount of algae, or the algal dynamics in the water column. So to summarize, it's clear from our experiment that there's this close association between methane production and algal dynamics in pelagic environments, and that this process represents an important fraction of total methane emissions from lakes. It's also clear that acetoclastic methanogenesis is important and likely increased in importance with increasing algal growth in the enclosures. And our results in combination with the meta-analysis suggest that the production and contribution of methane from oxic environments might shift from being a baseline source up to the main source as we shift from very small ecosystems up to very large ecosystems. So when we put this together, it's clear that from our results and the work of many others, that we need a better understanding of intercellular interactions in the microbial community at the cellular community level and subcellular level to get a better understanding of what's driving oxic water production and how the cells interact to support methane production in these oxic water environments. It's also clear that we really do need to account for methane production in these open water environments in future methane budgets. And because there is such a strong relationship between methane dynamics and methane emissions and algal dynamics, this really suggests that oxic water methane production could be linked with ongoing changes in algal dynamics linked to physical changes in lake habitats and trophic shifts linked to eutrophication and shifts in food web composition. So with that, I would like to thank everybody for listening. I really appreciate it. I also have to thank the Society of Canadian Oenologists and the CCFFR for giving me the opportunity to give this presentation and for the award. I thank my award nominators. I thank my co-authors and my supervisor, Paul Del Giorgio, for their support and contribution to this work. I also have to thank my lab mates for all of their contributions to this work as well. The Université de Montréal staff made this work possible. I want to thank my wife Holly for all of her support, as well as our funding sources. So thank you very, very much.
Thanks, Matt. So we'll move on to questions. There are 3 ways you can ask questions. For those of you who are participating to the Hangouts, meaning that you see your face at the bottom of the screen, you can mute and unmute yourself. As soon as you start speaking, your face will just come up on the screen. So that's number 1. Number 2, you can use the Twitter feed by following us. And on Twitter, just use the hashtag #LSeminar and ask your question. And so that's going to be easier for me to track you through the feed, so I can ask Matt, transmit Matt's your question. And finally, I think that if you're watching on YouTube, you can use the comment section on YouTube and just type your question. Pardon me, something I forgot to mention is that vous pouvez poser vos questions en français ou en anglais. Je serais heureux de traduire si jamais les questions sont posées seulement en français et si Matt ne... Je pense que Matt est capable de comprendre les questions en français de toute façon. So the floor is yours. Go ahead.
I have a question.
Yes.
Let me unmute. Now can you hear me?
Yeah.
Okay. So I'm really curious about this interaction between methanogens and phytoplankton, because would seem to be a very oxic environment. Do you think you have oxygen-tolerant methanogens in this system?
Yeah. So I think from what I've read in the literature that even in terrestrial environments, that the production of methane, that a number of methanogens are tolerant to oxygen. So this is one possibility that they're exposed to oxygen and they tolerate it, and methane production is ongoing despite that. But because the acetoclastic methanogens seem to be the dominant producers of methane, from what I've read, they're less tolerant to oxygen, and so I mean my thought is that maybe these micro anoxic zones are actually very important. So it's like at this point I'm not certain, but either the close association with algae makes very large micro anoxic zones that completely protect the methanogens from oxygen, or maybe that at the bacterial archaeal level there's interactions between the cells. I'm not sure, but yeah, I mean at this point I think I would suspect that these micro anoxic zones are protecting the cells from exposure to oxygen and allowing them to produce methane.
Do you have any plans to look into that any more deeply, or is this going to be somebody else's job down the road?
This is I had the same question than you, Julie. I'm really intrigued by this how they are physically organized in the water column.
Right. And it seems like this is the kind of thing that you could probably do with some nice fluorescence microscopy if you had a good bioimaging facility.
Yeah. No, totally.
If you need fish, you could use autofluorescence because those methanogens have something autofluorescent, don't they? It's kind of dim. If
Yeah.
you use them, I think you could find it.
Yeah. No, definitely you can image it. And I mean, it makes sense. We know that there exists a reduced sulfur cluster in the water column. There are stabilized metals. It's totally. Oh, it totally makes sense. Oh, good, you're back.
I'm not sure what happened. I've lost my connection a couple of times.
We just kept talking. So Julie had a good suggestion about I guess Julie's question was do you plan to look into this further?
If you're starting a postdoc, probably not.
I would love to look into it further at some point. I really think there are a number of levels that like with anything, this project we had I think a few insights, but a lot more questions came up from the work. So I think it would be really interesting to incorporate molecular level analyses and work with people that can do this, because it's a fascinating question, and I think it's a very important question, and I think that the different pathways are something that intrigue me, because when you look at the marine literature, there's an emphasis on this demethylation, and the one study that's looked at this in the German study that I made reference to suggests that the other pathways are likely very important in freshwater. So whether there's this kind of a difference between the environments or they actually align, I'd be very curious to explore this.
Yeah, well, I would imagine it's both. That there's contributions both from demethylation and from methanogenesis. And I really think that phytoplankton could be critical to both of those pathways, because those phytoplankton do also produce a ton of methylated small molecules.
Yes.
And so it's entirely possible that you would have correlation both of demethylation processes and of methanogenesis with phytoplankton. And so I think it's going to be really interesting to try and disentangle what other factors play into what's more important.
Yeah, I totally agree. And I mean, this is why my conclusion about exploring this at the cellular level and cell-cell interactions is why I think it's very important. Is this is the way to address the relative importance of these pathways. You can do a lot with the stable isotopes, but I think it would be best to combine this with the more local interactions.
Right. Yeah. Yeah. Thank you.
Yeah. Thank you.
Oh, I think we have a question on YouTube. So, Matt, do you think that anoxigenic phototrophic activity can contribute to a shift toward acetoclastic methanogenesis in anoxic environments by supplying acetate through CO2 fixation, assuming that other pathways, such as those dependent on hydrogen, are limited or absent? So, do you think there could be a link between some of those purple microbes, phototroph, enabling the environment to be conducive to such process? I guess this might be related to the phytoplankton or the phototrophic community, but which you identified as oxygenic, I believe. Although, just go ahead.
Sorry, can you repeat that? Just a quick abbreviation.
I can certainly repeat that. You think that anoxigenic photoautotrophic activity, so such, those anoxigenic bacterial photosynthesis, can contribute to a shift towards acetoclastic methanogenesis by supplying acetate that they would be providing through CO2 fixation?
Oh yeah, maybe. I mean, I don't know the answer to that question, but I mean, it's a great hypothesis, I think. I mean, the source of acetate is something that, like, my understanding is that acetate could be derived from many different processes and pathways, and yeah, I mean, I'm sure depending on the different lake environments, different sources of acetate could be important. So yeah, I mean, I wouldn't rule that out, and I would think that's a very important hypothesis to test.
Have you not in your enclosure studies, but in your review of the literature, have you noted that the presence of anoxigenic phototrophic microbes were somehow related to this oxic methane production?
No, I did not note that, and I did not actually look at the community composition and its relation to algal to methane concentrations.
Yeah.
But I think that's a very important next step.
Good. I think we are open to more questions. Anyone? Let me check. I don't have any here. Well, thanks everyone. This concludes our first live seminar. The link to that presentation is on our website, so you can just watch it over and over again. We welcome any contribution in French or in English. Please don't be shy. I think this is a wonderful, wonderful idea and concept. So thanks again to Micro Seminar and Phyto Seminar for bringing this up to our attention. This is a terrific opportunity for any early, mid, or late career scientist to broadcast their science from their office and for everyone to tune in without using massive and releasing massive amounts of CO2. So please you can contact me directly, Alex at UOttawa. You can contact the SCL through the SCL website, and you can just contact or direct message us on Twitter and let us know when and the title, and we'll get you started. So far, it's wide open. I've actually had a few requests for talks, so that's great. Thank you very much for putting your name forward, and thanks everyone. Good end of terms to everyone. Have a great spring, and hopefully see you at the end of April. Bye.